Continuing our discussion of Mendel, we want to look at what laws he started to apply to his work. We've talked previously about Mendel's crosses. This is showing you another example of a monohybrid or one trait cross. So we've already looked at the fact that Mendel was crossing a yellow flower or a yellow pea with a green pea and ending up with all yellow peas in his F1 generation, but in his F2 generation was ending up with yellow and green. So what we have to think about then is this idea of an allele. Please keep in mind that Mendel did not use the word allele. Okay? He knew nothing of genes or chromosomes or alleles, but what he did know is that two copies of something was being inherited from the parents. And depending on which copies were inherited, this could mask what was going on in his plants. So we talked briefly about the idea of dominant and recessive genes. We will talk more about those as we go through, but he reinforced the idea that alleles do not blend. So the traits that he is observing, and what we now know is meiosis is occurring to produce gametes, to pass on information to the next generation, is what we look at as basis for today's genetics. Now, in Mendel's peas, he looked a lot at these characteristics. This big P, and little p become important for us. Big P is dominant. Little p is recessive. This will become very, very important as we talk through things. But for now, we want to think about them as variables. They are alleles, as we know them today, and their variation is important for giving differences to the phenotypes of organisms. As far as inheritance of these alleles, the organism could inherit from this, in this case, only big P's, right? This parent is giving only big P or big P to its next generation. This individual is giving only little p or little p to the next generation. Fine. But when you get down here, you see this individual, even though it's purple in its phenotype, has a genotype that is big P, little p. That means that if those alleles would separate, those chromosomes separate in meiosis, one is going to go to one gamete, one is going to go to another gamete, and depending on the gamete that is inherited, that will determine what characteristic that next generation, the F2 generation, is going to have. So we'll look at this in more detail, but we call this genotype, right? This big P, little p actually gives us our genotype. So when we look at those alleles, we're talking genotypes. When we're looking at traits or characteristics, or in this case colors, we look at phenotype. So in our F2 generation, we can start thinking about the idea that we're getting this 3-to-1 ratio that Mendel often looks at, or 75% to 25% in color. But if we actually look at the genotype, we have one that's big P, two that are big P, and one that's little p. What does this mean in the long run? Well, we have to make sure that we are very clear when we talk if we're talking about a phenotypic or genotypic ratio. Okay? You must be clear on this. They are different in most cases. So Mendel came up with a law known as the Law of Segregation, saying that these alleles that we talked about, big P and little p, separate before forming the gametes. Mendel didn't know it, but what he was describing is meiosis. It fits perfectly with what we know in genetics today, and he had no idea. So this phenotypic and genotypic ratio that I was just mentioning to you will be important when we consider how these different crosses can result in highly variable offspring. That variation will be very important for us later when we talk evolution. Without variation, there is no evolution. Okay? So we'll come back to that and think about it more.